about, you know a lot about the heart. Now we're going to add the vascular system into this and really finish today the lecture material about the cardiovascular system. The blood vessels that you're learning the anatomy of are not just tubes. They are part of the anatomical and physiological system of the cardiovascular system. So we're going to learn about how the cardiovascular system, both the heart and the vascular system, regulates blood pressure and blood flow. That's where we're going. So you know, this one you already know. This one we have covered. How the sympathetic nervous system increases the heart rate by the mechanisms that we covered on Monday, and how the parasympathetic nervous system slows the heart rate. And we can talk about that during the breakout if you have questions. We're going to talk about how the strength of the heart is regulated and how that controls something called stroke volume. Stroke volume is defined by the volume of blood that is pumped out each beat. So each beat, each contraction pumps a certain volume of blood, and that's the stroke volume. At the end of diastole, that is to say at the beginning of systole, the very beginning of systole, there is a volume in the heart. The heart is completely refilled, and that's called the end diastolic volume. So there's a volume at the end of diastole, at the end of refilling. There's a volume at the end of systole, when it's at its minimum, and the stroke volume is the difference between those two volumes. We will talk about a concept called cardiac output. Cardiac output is the amount of blood the heart pumps per minute. So it's a product of the number of beats per minute times the volume per beat. Pretty straightforward arithmetic. And then finally, we're going to get to the concept of blood pressure, how the heart and the vascular system generates pressure, and we'll get another equation related to how resistance of the vessels and output of the heart generates a pressure. That's where we're going today. And so let's talk, as long as we're talking about pressure, let's talk a little bit more about pressure. Maintaining pressure is one of the most important functions of the cardiovascular system. Because without pressure, you don't have flow. And without flow, you don't have the blood going to where it needs to go. So we think in terms of flow, but it's also useful to think of pressure because pressure generates flow. So maintaining blood pressure is one of the most important functions of the cardiovascular system. Pressure, so some of you might remember Ohm's law from electricity. Well, this is Ohm's law, but for fluid instead of electricity. Pressure is flow rate times resistance. Ohm's law for electricity, for those of you who know it, is V equals I, a flow of electricity, times resistance. So if it helps you to draw the analogy to electrical resistance and Ohm's law, pressure is equivalent to voltage, current is equivalent to flow rate, and resistance is resistance in both circuits. So the flow rate that we're going to be talking about is the cardiac output, volume per mils per minute. The resistance we're going to talk about is the resistance of the system that the flow is going through. That's the systemic vascular resistance. So resistance is the resistance of the system. Sometimes it's called total vascular resistance. That makes sense too. So the output of the heart multiplied by the resistance of the vessels that the flow is going through generates pressure, and we call it the mean arterial pressure. mean just meaning average. Okay, and remember, so cardiac output is the rate in beats per minute times the stroke volume, volume per beat. So that becomes volume per minute. And so if pressure is rate times resistance, well, here is rate, heart rate times stroke volume, that's cardiac output, and there's resistance to generate an equation for pressure. And I'm not going to ask you to do a lot of arithmetic with this, but I think having these equations helps you think formally about, you know, what is changing when we change, you know, if we have hypertension, high blood pressure. Is it because we have an abnormality in heart rate, or an abnormality in stroke volume, or an abnormality in resistance, or all three, or two of the three, you know? And so if we have low blood pressure, why? So it allows you to sort of parse out what is, you know, the pieces that contribute to things like high or low blood pressure. So here's a flow chart figure from your textbook. It's the end point here that we're trying to understand is cardiac output. So cardiac output is stroke volume times heart rate. So it's looking at heart rate and stroke volume are the inputs to cardiac output. So you already know that the sympathetic nervous system, norepinephrine from the sympathetic nervous system, and epinephrine from the adrenal gland, which is really a part of the sympathetic nervous system. So norepinephrine and epinephrine activating beta-1 adrenergic receptors in the heart, GS, the whole story we talked about how that increases heart rate. You should be able to tell that story, and if not right now, pretty soon. So I'm not going to do this again unless you ask me to do it again, because we've done it. Epinephrine and norepinephrine increase the heart rate. Beta-1 receptors, sympathetic nervous system, GS, increasing, and I'll give you the outline, increases IH, increases calcium channel activity, increases the closing of K channels, increases the pacemaker rate of depolarization of SA node, which increases heart rate. Okay, I went through that fast, but I think I've gone through that. What we're going to do now is talk about how norepinephrine and epinephrine increases stroke volume. That's what we're going to do right now. And here's the slide that has the story embedded in it. So let's walk through the story. This will be helpful actually for the rate part too, because some of it is the same story. So let's walk through it. I need a verb. What do these things do here?
Bind.
Binds, good. Then what? Conformational change. What happens to the G protein? Okay, conformational change, and then dissociation of GDP from alpha and binding of GTP to alpha. If you say ATP here, you're wrong and you won't get full credit. Yes?
For the problem set for the last question, do we have to go in such detail for the story?
For the what?
For the story. So for example, when we say the epinephrine, norepinephrine, do the GS story, do we go through the whole, every step of GS?
If I ask you for cellular and molecular mechanisms, and you say GS story, I'm going to say, yeah, more, I need more.
Okay.
Okay? If I tell you, you do not have to go through the GS story, or you do not have to go through the cross-bridge story, then you don't. But if I don't tell you, you want to give me everything you got.
Okay.
Do you want us to know where more epinephrine and epinephrine come from?
that's really ANP1. So the short answer is no. I'm not going to ask you about sympathetic nervous system anatomy and physiology. I'm not going to ask you about the adrenal gland, which is the source of epinephrine. So we're just black boxing the sympathetic nervous system and the adrenal gland in this course. If you want to know more about that, take ANP1. Yeah.
This might be a dumb question, but are norepinephrine and epinephrine, aren't they allowed to pass through the membrane?
No. Epinephrine and norepinephrine are both too hydrophobic to pass through the membrane. So where were we? GTP binds to alpha. Alpha separates from the receptor and from beta gamma. So this is the activated alpha. Okay, what happens next?
That
Activates adenylyl cyclics, which
increases
converts ATP to cyclic AMP, which, what is this arrow illustrating? Diffusion. I think I heard you say that. Yep. Diffuses in the cytoplasm and binds to, what does it bind to? So this slide has missing information, as they always do. What does it bind to activate PKA?
Tyrosine
It binds to what?
and the side chains?
Well, yeah, but I'm thinking about in terms of whole protein, the regulatory subunits. So cyclic AMP binds to the regulatory subunits, which liberates the catalytic subunits of PKA. It activates it. And so if you just say cyclic AMP activates PKA, I'm going to say, well, yes, it does that, but we've talked about the mechanism. It binds to regulatory subunits, conformational change, of course, and the regulatory subunits diffuse away, liberating catalytic subunits of PKA. What does PKA here? And here are the catalytic subunits of PKA. Okay, now what is PKA doing in this system? This is a we're in a cardiac myocyte. We're trying to get to understanding increases in stroke volume. So what does PKA do? What is this trying to illustrate?
increases the activity of calcium channels and IH
By,
channels.
well, this isn't IH. So this is voltage-gated calcium channels. It increases their activity by doing what to them? What do kinases do to proteins? Phosphorylates the voltage-gated calcium channel and increases its open probability, increases its ability to open. More calcium influx. Okay, let's just hold that story right there. What is PKA doing here? It's phosphorylating what? What is this trying to illustrate?
The calcium
Yeah, it
release.
says it right there. The calcium release channel in the SR. So now the calcium from the voltage-gated calcium channel and phosphorylation from PKA increases calcium release from the SR. What does this calcium and this calcium do? What is this trying to illustrate here? What is it binding to? Troponin. Now we're off and running with the cross-bridge cycle. If I say, you don't have to tell me about the cross-bridge cycle, then you don't. If I don't say it, you do. You would give me the whole shtick about cross-bridge. you tell me the story of the cross-bridge cycle, because that's a cellular or molecular or both mechanism. Okay, cross-bridge cycle. More cross-bridge cycling, more contraction, more force being generated, more pressure being generated by the heart, by the ventricles. Okay, so we've gotten to the story of increased in the force and speed of contraction. Okay, this is the last part of the story. What is happening here? what is this illustrating, this calcium illustrating? Reuptake. Is that happening during contraction? It's happening during relaxation. But remember, every contraction has to be followed by a relaxation for there to be another heartbeat. And so this is an important part of the play. But what this slide doesn't show is that you should draw this arrow into your slide. pKa also phosphorylates a subunit of the circa and makes it pump faster. So that's absent from the slide, but that's an important part of what pKa does in a ventricular myocyte. So you can say, you could annotate your slide at the bottom. It increases the force and speed of contraction, and it increases the speed of relaxation, which is shown in the next slide. Sympathetic, red is sympathetic, green is control. We're looking at force of a muscle cell as a function of time. Control, there's calcium influx and it generates force of contraction. When stimulated by the sympathetic nervous system, we have an increased speed of contraction, an increased peak of contractile force, and an increased speed of relaxation. More contraction, stronger heartbeat, more stroke volume. Okay? We're part of the way through understanding the change in stroke volume. Questions about this story? This slide has a lot of information, and I told you even more information about it, the regulatory subunits and pKa phosphorylating this. So that's a story you should tell yourself in order to understand it. Okay. Now we want to take another piece of this flow chart and understand what's going on here. Why does the volume at the end of diastole affect the volume that is pumped by the heart? That's another part of this flow. We just did these two arrows, how the sympathetic nervous system, epinephrine and norepinephrine do this. Now we're trying to understand this arrow. The fact is, so let me just show you the data first. If we plot stroke volume as a function of end diastolic volume, there is a relationship with an increase. Up to, it doesn't keep going, it maxes out. So at rest, your stroke movement is about 70 mils. But when stimulated by a number of things, and we'll get to that in a minute, your stroke volume increases because of the sympathetic nervous system, but also just because it is getting filled more. We'll talk about why in a minute, why it gets filled more in a minute. Right now we're trying to understand this increase. The why it increases in contractile force as it increases in filling volume is shown here. So let's parse out this slide. Okay, so these cartoons are supposed to be illustrating a sarcomere of a ventricular myocyte. If the sarcomere is sort of perfectly situated, the head groups of the myosin are all perfectly in position with the thin filaments, with the actin of the thin filaments. Every single head group has a place to bind on a thin filament. And so that generates a perfect, the best contraction this muscle can generate. If the muscle is not stretched to a perfect length, so say it's only stretched to this amount, this is an exaggeration, but you could see if this sarcomere was a little bit shorter, then there's going to be head groups, well, but there's going to be some interaction between the thin filaments. And these head groups of the thick filaments are not perfectly situated for interacting with thin filaments, and the thin filaments are actually getting in each other's way. And so there is less force when you have a less than perfect overlap of thick and thin filaments. And so this is our situation at rest. It's not perfect. We can move, actually, the heart works somewhere down here. We can move up this curve by improving the thick and thin filament geometry.
Would you call that an alignment?
Well, it's not exactly alignment. Overlap is the better word. Perfect overlap of thick filament head groups and thin filament binding sites. And here it's imperfect. So lower force, maximum force, and in fact, if the sarcomeres are overstretched, you can see there are head groups that can't bind to a thin filament. And so again, if it gets too stretched, the strength falls down. So going back one slide, if we go out here, it actually starts to fall off very suddenly. So so we are normally working here. This is a healthy range. In the case of dilated cardiomyopathy, which is a nearly always fatal heart disease, the end diastolic volume gets out here, and we get into a catastrophic decrease in cardiac output. That's heart failure. Yeah.
So I guess the overlap of the thin and thick filaments, is it associated with calcium that is present in the cytoplasm, or?
This is independent of calcium. This does not depend on calcium. This is the situation at rest. So at rest, these thick and thin filaments are perfectly situated to contract with the maximum force. At rest, these are not, and at rest, these are not because of this bumping up together of the thin filaments from the two two Z discs. Sophie?
So what determines the length of a muscle?
What determines the length of the muscle is, in the heart, is the amount of blood that has refilled it and stretched it out. So in order for skeletal muscle to stretch, you need another muscle to stretch it. For heart muscle to stretch, you need blood to stretch it. Okay? So 100% is perfect. So 100%, this is perfect stretching. And this is less than perfect stretching, and this is overstretching.
The previous graph for x-axis had the end diastolic volume.
Yeah.
Is that showing, so then when there's too much blood, it's stretching it too much, and that's why we plateau and we plateau?
Too much blood would be out here.
Okay.
So the heart, the point is we have reserve, we can stretch it more and generate more cardiac output because the perfect amount of stretching is actually quite a bit more than what we typically use. We've got a reserve here that we can stretch it more and generate more force. But when you get out here, you got to be careful because you're kind of close to the fall. So we typically work in this range, which means on this graph, we typically work in this range. Yeah,
So like, are you at 100% on your exercise?
yeah, pretty close. A healthy person exercising is generating pretty close to the maximum cardiac output. I mean, someone really exercising. So now the question is, so now I think I've explained why we can increase the force when we increase the stretch due to increasing the volume that has refilled the heart. Okay, so I've explained this change. Now I need to explain, well, how can there be more volume returning to the heart? How can we get more refilling? How can we get a larger or smaller end diastolic volume? And the answer to that means going back to this slide from last week, where I told you that about 3/4 of the blood is in the low pressure side of the heart, of the circulatory system. I don't think, did I print this one out for you? I don't think so. So the point is, 3/4 of the blood in your body at any given instant in time is in the venous, the systemic venous and the pulmonary venous circulation. Only about 1/4 of the blood at any given time is in the systemic arterial and the pulmonary arterial system. So the venous system, and particularly the systemic venous system, this right part here, is a reservoir of blood. It's where 3/4 of the blood is. It's under low pressure, high volume. If we constrict the smooth muscle of the veins, we reduce the volume of the systemic venous circulation, which means more blood has to go somewhere, and it gets back to the heart. So constriction of the veins shifts the blood into the heart and into the systemic arterial circulation. So what I just showed you is that by constricting the smooth muscle of the veins, we increase return, which increases end diastolic volume. Okay, so increasing end diastolic volume in the right heart, increasing end diastolic volume in the left heart, and now we have generated the reason why the end diastolic volume can increase. And of course, if we relax the vascular smooth muscle of the systemic veins, we can reverse it. So constriction of the systemic venous smooth muscle forces blood to return to the heart, reduces the reservoir, and increases end diastolic volume, the volume at the end of diastole. No?
So with the inverse relationship of pressure and volume, wouldn't constricting the veins actually cause less blood to be delivered? Because high pressure means it's constricted, so less blood can pass through the veins.
There is more resistance, but there is also more return.
Is that the cardiac output?
The thing that controls resistance is not going to be the veins. We are going to control resistance upstream of the veins. So that's going to lead you down the wrong path.
That makes sense.
Question, Pradesha?
I was just asking if that, the flow then would be the cardiac output. If the resistance is higher, but also the cardiac output would be higher, right?
If filling is higher, stroke volume is higher, and cardiac output is higher. So we just explained why more filling, more end diastolic volume leads to more stroke volume. We just explained this arrow. And this part of the slide is simply illustrating that veins have smooth muscle. And so smooth, circular smooth muscle is going to constrict the veins. Questions before I move on? All right, so I'm switching gears a little bit. I'm now going to explain why the pressure in the aorta stays high even when the pressure in the left ventricle has fallen to nearly zero. So remember, this is your Wigger's diagram. Here is systole from the beginning of isovolumetric contraction to the beginning of isovolumetric relaxation. Ventricular pressure goes from very low to very high and to very low again during contraction and relaxation. But aortic pressure never gets very low. Why is that? That's where we're going. And the answer, this is just a cartoon of an artery, and it shows structure. We'll talk about this more in the second hour lecture. But the point is, arteries, and veins for that matter, but now we're talking about arteries, because the aorta is an artery, are elastic. They are stretchy. You pump a volume into them, and they stretch. They have elasticity, and for physiology, we talk about that as compliance. Arteries, even the aorta, which is pretty thick, is pretty elastic. But when we get to major arteries like the brachial artery, or the radial or the ulnar artery, or the femoral artery, major arteries, those are super stretchy. They are elastic. So you pump a volume into them, and they stretch. The stretchier so now getting into some biophysics, I'm not going to do this in great detail, but the point is that a thing called compliance, and you in your mind can say, more compliance means more elasticity, means more stretchiness. More compliance means a greater change in volume for a given change in pressure. That makes sense. More volume for a pressure means more stretchy, which is more compliance. No?
Does that mean arteries have more sarcomeres?
Or what?
Arteries have more sarcomeres to be more elastic?
Well, arterial smooth muscle, no, I don't think that's fair to say. Arterial smooth muscle actually is thicker than venous smooth muscle, but there is still plenty of stretchiness. Both of them, both arteries and veins, are compliant. In fact, veins are more compliant, but now we don't have to worry about vein compliance here because we're talking about arterial compliance. So veins are plenty compliant, but that's not what we're talking about. We're talking about arterial compliance. So what is this cartoon supposed to illustrate? This is supposed to be illustrating arteries or the aorta. This is a valve. So what valve pumps blood into the arterial system? Or no, what part of the heart? What part of the heart is doing this systole, pumping blood into the arteries? Left ventricle. What valve is this supposed to be illustrating?
The aortic semilunar valve.
The aortic semilunar valve. You have to sort of turn these cartoons into what you know. This is the aortic semilunar valve. This is the left ventricle, pumping blood into the arteries. The arteries are stretchy. So during systole, the arteries stretch because of their compliance. And during diastole, when there is no more pressure being forced into them, the stretchiness relaxes. It's just like you blow a balloon up, and then you let it go and the air flows out. You pump a balloon up, then you stop blowing and let go. Well, there's the let go. The stretchiness is maintaining the pressure between systoles. So we just explained, at least for now, why arterial and aortic pressure stay high even when ventricular pressure is low. It's because the aorta and the arteries are compliant. They stretch during systole, and then the elasticity maintains pressure and flow during diastole.
So why is there, like, logically, my hand's not doing it right, but like, why is there more pressure when you're stretching? Like, wouldn't, when something's smaller, there be more pressure going through it?
Well, it's the pressure that's stretching. So this pressure from the heart is stretching the arteries.
So wouldn't they have lower pressure overall since they're stretched?
Yeah, but this is a big force, maintaining pressure. So yeah, I mean, if this were to stretch infinitely, yeah, this would become low pressure. But obviously it doesn't go that far.
So that's why the pressure remains, okay.
So it's, if your arteries don't stretch, then the pressure from the ventricle will cause, all of that pressure will get transmitted into the arteries and you have hypertension. That's high blood pressure. Hardening of the arteries generates hypertension because the arteries don't stretch. So your parents and your grandparents, your professor, has harder arteries than you do and probably higher overall blood pressure because of the lower compliance of the circulature, of the vasculature, the arteries in particular, in elderly people.
So why don't the veins have, like, could the veins have high blood pressure as well, since they don't stretch?
We don't worry about hypertension in the veins very much.
Okay.
It's arterial hypertension that is the health problem. So I transitioned into a health topic, but I mean, it helps to make it make sense. Is this story then clear? We're explaining why this pressure stays high and why there is continuing blood flow even during the time when the heart is relaxing. Okay, so now we're going to talk about the circulatory system in some detail. So this is just a sort of a flow chart, right? Aorta. So we're going to start at the biggest artery, and we're going to end at the biggest vein. Biggest artery. Major arteries that you are learning the names of and will be on the quiz. More major arteries. We can just lump these together. But eventually these major arteries divide into smaller arteries that are called arterioles. And they divide and they divide and they divide. These are small to microscopic, and they don't have names. And then they divide further into capillaries, tiny little blood vessels, where nutrient exchange happens. And then those venules that are small or microscopic and don't have names, major veins that do have names, and that's, some of those are names that you're learning. And then the inferior and superior vena cava. Okay, so that's just reminding you that there is division. And here's a slide that illustrates it. So here is this is an artery. So here is a primary, let's just say this is a primary arteriole. This is a micrograph. So here are the capillaries, tiny little things. Here's an arteriole, branches, Let's follow this branch. Branches, let's follow this branch. Branches, 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 network of capillaries. Okay, so this slide makes sense. The arterioles are where blood pressure and blood flow are controlled. Why? Well, you'll see. The arterioles are the most important part of the circulation for controlling systemic vascular resistance. So remember the equation. Mean arterial pressure, which is just blood pressure, is equal to cardiac output times systemic vascular resistance. And what I'm telling you is that the resistance, the most important part of the resistance, is at the arterioles. The resistance of the arteries, the resistance of the aorta are not as important as the resistance of the arterioles. That's where we control systemic vascular resistance. We're going to talk about that more after the break, after the breakout session. But not only is the arterioles where systemic vascular resistance is controlled, it's where the blood goes. We have variable control of where the blood goes. So this is supposed to be, like a reservoir of arterial blood. And this is supposed to illustrate a set of arterioles. And these are supposed to illustrate different parts of the body. So let's just say one is, let's see, one doesn't change, so let's just say this is kidney. Some fraction of the blood goes to the kidney. And this is, let's say this is skin. I'm just making this up. And let's say this is skeletal muscle. And let's say this is some other part of the body, digestive tract. And this is some other part of the body. Brain, you want brain again? We already did brain, but whatever. Brain, skin, digestive tract, kidney, muscle, liver, the places where the blood goes is controlled. Where it goes is determined by the constriction or dilation of the arterioles that feed blood to that organ. We have specific control. So that's the variable resistance, variable control of blood to those organs. And so let's just say this one goes from fully dilated and gets a lot of blood to fully constricted and gets very little blood. And this one gets a lot of blood and gets a lot of blood no matter what. So this one is a pretty good illustration of the brain. The brain gets a lot of blood no matter what. Here is a pretty good, this one, number 3, is a pretty good example of what happens to skeletal muscle during exercise. During exercise, the blood goes to the skeletal muscle. And it also goes to the brain. But it doesn't go, let's just say here, when you exercise, blood is diverted from your digestive tract and your kidneys to your skeletal muscle. When you exercise, you do not need blood going to your digestive tract. That's why they say don't eat something immediately before exercising. You don't want a bunch of food your digestive tract to try to be digesting a bunch of food when when the blood flow is going to be limited because it's now being diverted to the skeletal muscle. We do that by specifically constricting the arteriolar smooth muscle of the vasculature that leads to that particular organ. We're going to talk more about that after the breakout session. Some circulatory beds get more blood under certain circumstances, and others get less blood. So where does the blood go when you have a big meal? It goes to the digestive tract. Where does it not go? Skeletal muscle. Where does it go no matter what? I think I told you this. Brain. So there are some circulatory beds that are under less control and some circulatory beds that are under more control, whether you're digesting or exercising or sleeping, etc.
What about kidneys?
Kidneys,
So sorry, but
kidneys they demand their blood pretty much no matter what. Well, that's not true. No, that's not true. When you're exercising, the blood flow to the kidneys decreases because we don't need to be generating a lot of urine when we're exercising. Sweating, so skin, it's interesting. Skin is interesting, so when you immediately start exercising, blood flow to the skin is decreased. And you may have had the experience of when you start working out, your skin, you feel cold. But then your body heat, your muscles start generating a lot of heat, and you start sweating. And then under those circumstances where now we have a thermal control, high body temperature tells the system, oh, we need to get rid of this excess heat. Now we need to dilate blood vessels to the skin, and in order to get rid of extra heat by sweating and by sweating, evaporation, and radiation of heat. So exercise causes the constriction and then the dilation of blood vessels to the skin. So does this ring true? I mean, it certainly happens to me. Within 5 minutes of exercising, my thin skin feels cold, and then maybe 5 minutes later, I start to sweat and it feels opposite. Do you have the same? Have you noticed that in yourself as well?
I usually start to sweat, or I usually start to feel cold because I start to sweat.
Oh, so the sweating actually chills you to the point where you start to feel cold. I don't.
I'm prone to feeling cold all the time.
Oh.
So, that might be possible.
Okay. Yeah.
Why does the skin, or why does your body decrease blood flow to the skin?
It's diverting the blood to the skeletal muscles. It's taking it from where it can get it, and the skin is a pretty good source. So by constricting blood flow to the skin, the body is saying, okay, I don't need to get rid of a lot of heat, but I am exercising, so I need to get this blood to the skeletal muscle that is working out. So skin is one of the first things to get constricted so that there is blood to go to the skeletal muscle that is dilated. And then the body starts to realize, oh wait, I'm getting hot. I need to dilate the blood vessels to the skin, and now it constricts it to the liver or the kidney or something else. It's a pretty smart thing, this body. So finally, before we take a break, here is blood pressure. You've had your blood pressure taken. You know that you've got a number 120/80 or something. Here is the 120/80. So 120 is the millimeters of mercury of pressure. So you could, did I give you a handout of this? So the units, oh there it is, millimeters of mercury is the units of pressure in physiology. So maximum systolic arterial blood pressure is the high number, 120. No, typical. 80 is the low systemic. So 120, well, so it doesn't really show it, but there, 110 over 7. That's nice, healthy, low blood pressure. 120 over 8. A little high for someone your age, pretty good for someone my age. Systolic, diastolic, systolic, diastolic. Each of these is a heartbeat. This is what's happening in the arteries. As we move the blood through the arterioles, remember their arterioles is where the resistance is. And so the pressure, I mean, we're still getting oscillations, but on average, the pressure is dropping over the arterioles because of resistance. So there's not very much resistance in the major arteries. There's a lot of resistance in the arterioles. There's some resistance in the capillaries. And by the time we get to the veins and venules, the oscillations have been sort of blotted out. So you may know that, you know, if you cut an artery, you get spurts of blood. But if you cut a vein, you just get blood flow. Lower blood pressures in the pulmonary circulation. You still get diastolic and systolic, but lower pressures because the right ventricle is so much less meaty than the left ventricle and generates much lower pressures. But the same concept, the arterioles and the capillaries is where the resistance is in the pulmonary circulation. So this is why we don't have to worry about resistance in the vanules and the veins, because it's the pressure has already dropped in the arteriolar system and in the capillaries. Your book has a description of how these pressure waves going from 80 to 120 generate the sounds that we hear when you do a blood pressure measurement. I think your book does a really good job of explaining it. It has to do with flow and turbulence. All you need to remember is that smooth flow is silent, no flow is silent, smooth flow is silent, but turbulent flow makes a noise. And that's what we listen to. And that's what those automatic blood pressure cuffs are listening for, a noise. So rather than explain it, because I'd like to stop, I'm going to let you learn this from the book or from each other during the breakout. So what I would like to do now, answer any questions that you might have now, but then take a 15-minute break, go to your Zoom room, Zoom location, join the breakout Zoom, which is on Moodle, and we'll start working on problems, whether it's the problem from the problem set or a bunch of problems that are on this PowerPoint. So have the PowerPoint in front of you on your screen, have the problem set on either on your screen or on a piece of paper in front of you, and then you will work with your classmates on which problem you think is most important. So let's just go through what's on here. So here are some images of the heart, and you might want to annotate them with labels. So we saw this in the lab, something that looks an awful lot like this. Here is an posterior superior view of the heart. So you can, and this is just this heart sort of, this is the same picture of rotating. So you could get oriented and label this one. Here are the valves of the heart, looking down at the ventricles with the atria having been cut away. So and this is right and this is left, I think. So you could if I were to give you this picture and ask you to label it, I'd have to tell you which is left and which is right. So, do some anatomy together. Or do these questions related to the Wiggers diagram? Or answer this question about acetylcholine and how acetylcholine causes a decrease in blood pressure. This is sort of the reverse of one of the questions on your problem set. The last problem on your problem set is to describe the molecular mechanisms for the sympathetic nervous system. Here's another one, but this is looking at the parasympathetic nervous system. Answer the question, where is the highest resistance? I think I just answered that. And then here is arteries. So I think that I printed this one out for you, but they didn't realize that when in color they look different. In black and white they look almost exactly the same. But the first one is arteries, and the second one is veins. And work together on trying to label, basically take the information from your brain that you learned from the coloring book and map it onto a different picture as a way of reinforcing your knowledge for the quiz. And then there's a boatload of clickers. So what I'd like you to do, I'll stop talking in a few minutes, unless you ask me questions. We'll take a 15-minute break, and then I'll make notice of the time. And that's when we'll start the Zoom class and then go into breakouts. Your job during the breakout, let me just be clear, your job during the breakout is to just, work with your classmates on, okay, I'd really like to talk about question 9 on the problem set. Or I'd really like to talk about the anatomy problem. We'll do this for a good 45 minutes. So you have plenty of time to work on a lot of things. And yes, so
For, when you say isovolumetric ventricular contraction, does that include like the blood leaving the ventricles?
what do you think?
No?
No. Isovolumetric means the volume is not changing.
Okay.
If the volume is not changing, it's not leaving. And so blood ejection is not part of isovolumetric. And blood refilling is not part of isovolumetric relaxation. Isovolumetric contraction and relaxation means there's contraction, but all valves are closed. So there's no blood flow. Isovolumetric relaxation means there's relaxation, but all valves are closed, so there's no refilling. It only lasts for 50 milliseconds, because eventually, pretty soon, a valve is going to open, and you're either going to get ejection or refill. Okay, so it is. Sophie?
Does the QRS complex like involve the like opening and closing of the valves?
Well,
Because it is like the contraction, but does it also involve like what's going out?
so we got to be clear here. The QRS complex is not really associated with contraction or relaxation. The QRS complex is a manifestation of electrical activity in the heart. But the QRS complex is most closely associated with the onset of contraction. Did that get it, your question? The onset of relaxation is associated with the T wave. No?
Just to be clear, what I'm understanding, we're not talking about preganglion neuron and postganglion neuron, we're talking about sympathetic nervous control of the blood pumping. How
We're talking about, so this is AMP1, we're talking about postganglionic neuron release. So we're talking about release of norepinephrine from the postganglionic sympathetic neurons, and we're talking about ACH release from the postganglionic parasympathetic neurons. We are not talking about anything pre-ganglionic.
Yeah, so